pleasant morning, everyone. So, how are you guys? Well, I know some of you have your eyes almost closed back since we're starting our class quite early. Am I right? So, before going into our formal discussion, our group, the Visionaries, will make your eyes open first and activate your brains with these amazing optical illusions. Hope you guys enjoy it. Here we go. After having one look at this optical illusion, most people say that the circles are moving in different directions. If you move your head from side to side, you might even get a little dizzy. But in reality, the circles don't actually move. How many faces are there? Most people initially notice a girl in profile with their head turns toward the viewers. But if you look more attentively, you'll realize that you're seeing just one half of her face while the other is in darkness. Are these men the same size? Almost anybody who looks at this image will be 100% convinced that the men's silhouettes are gradually getting bigger from the bottom to the top. However, that's just an optical illusion created with the help of divergent lines. All of the men are the same exact size. Can you guys read between the lines? Hopefully, 3 seconds has been enough for you to spot the word love written across the image. It is indeed hard to read since the letters blend so well into the background. Can you see a sleeping woman in the picture? This one seems impossible, right? There's just an old man sleeping peacefully in his bed. No woman inside here. Let's add some color to help you out. Viola! There's the brown-haired young lady. She's just sleeping on her side and turned away from the viewer. And that's it! Those optical illusions were amazing, aren't they? And while we were watching, trying to figure out what was exactly happening with those images, we know that our eyes were the busiest. Widening them or squinting at times just to focus on the image for our brain to process it. Oh, our hardworking eyes. Thinking all about this right now, aren't you guys curious of how our eyes really work? Aren't you curious of the process of our vision? Well, if everyone's curious, then worry no more. Because today, I, together with my groupmates Crystal, Drake, and Andromeda, aka the Visionaries, will do our best to discuss the visual system and basically explain how we see. So, let's dive in. Complete darkness. Humans cannot see in this state. But what about cats, owls, and other nocturnal animals? Do you guys think they can see something in complete darkness? The answer is no. Some animals may have special adaptations that allow them to see under very dim illumination, but no animal can see in complete darkness. In essence, if there is no light, there is no vision. The light reflected into our eyes from the objects around us is the basis for our ability to see them. Light can be thought of in 2 different ways: as waves of energy or as discrete particles of energy, which we called photon. Both perception and light are useful, because in some ways, light behaves like particles, and in others, It behaves like waves. Physicists have learned to live with this nagging inconsistency, and I guess we should do the same. Light, or visible light, refers to the region or segment of the electromagnetic spectrum that the human eye can view. So, as you can see here, visible light is only a certain part of the spectrum. We see light between 380 to 760 nanometers. That is visible light. We cannot see gamma rays, nor infrared rays. But other species have slightly different ranges from us, meaning they can see wavelengths that we cannot. For example, rattlesnakes. Rattlesnakes can see infrared waves, which are too long for humans to see. As a result, they see warm-blooded prey in what for us would be complete darkness. But this specific range here is ours. And within this range, the specific wavelengths gives us the perception of color, and intensity gives the perception of brightness. These two are the properties of light. So how does light enter the eye? Light rays bounce off an object we are looking at. Let's say the object is a dog. The light reflects off the dog to our eye. Then the light enters through the protective outer layer of the eye called the cornea. The cornea is clear like a window. It helps our eye focus the light to make things look sharp and clear. Next, the light rays pass through an opening or hole called pupil. The pupil is the dark round circle in the colored part of the eye that allows light to strike the retina. Meanwhile, this colored part is called the iris, which gives our eyes the characteristic color. It also controls the amount of light that enters the eye by changing the size of the pupil. This adjustment of pupil size represents a compromise between sensitivity, or the ability to detect the presence of dimly lit objects, and acuity, the ability to see the details of objects. In bright light, when the level of illumination is high, the sensitivity is not important. Thus, the visual system takes advantage of the situation by narrowing a pupil by the iris, reducing the amount of light that enters the eye. This is called constriction. When the pupils are constricted, the image falling on each retina is sharper and there is a greater depth of focus. In dim light, when the level of illumination is too low to adequately activate the receptors, the iris widens the pupil to let in more light. This is what we call dilation. Here, acuity and depth of focus is sacrificed. Behind the pupil sits the lens. The lens focuses the incoming light on the retina. And what moves this lens back and forth are ciliary muscles. Ciliary muscles alter the shape of the lens so that we can see things up close and far away as well. When we direct our gaze at something near, the tension on the ligaments holding each lens in place is adjusted by the ciliary muscle, and the lens assumes its natural cylindrical shape. This increases the ability of the lens to refract or bend light and thus brings close objects into sharp focus. When we focus on a distant object, the lens is flattened. The process of adjusting the configuration of the lenses to bring images into focus on the retina is called accommodation. The movements of our eyes are coordinated by converging or turning slightly inward so that each point in our visual world is projected to corresponding points on our two retinas. But the positions of the images on our two retinas can never correspond exactly because our two eyes to not view the world from exactly the same position. Binocular disparity. Sounds familiar, right? Binocular disparity is the difference in the position of a particular image on the two retinas. The greater the disparity, the closer the object is. So having two eyes is what gives us our depth perception. Before proceeding, let's talk a bit in an evolutionary perspective. No description of the eyes of vertebrates would be complete without the discussion of their most obvious feature, the fact that they come in pairs. One reason vertebrates have two eyes is that vertebrates have two sides, the left and the right. By having one eye on each side, which is by far the most common arrangement, vertebrates can see in almost every direction without moving their heads. Here, you see three animals, a human, an owl, and a lion, whose eyes are on the front of their heads, and other three, an antelope, a canary, and a squirrel, whose eyes are on the sides of their heads. In general, predators tend to have the front-facing eyes because this enables them to accurately perceive how far away prey animals are. It's the basis for the visual system's ability to create three-dimensional perceptions or to see depth from two-dimensional retinal images. While prey animals tend to have side-facing eyes because this gives them a larger field of vision and the ability to see predators approaching from most directions. After light passes through the pupil and the lens, it reaches the retina. The retina converts light to neural signals, conducts them toward the central nervous system, and participates in the processing of the signals. The process by which light hitting the retina is converted into neural signal is quite complex to say the least. To begin to understand this, let's zoom in a bit on the retina. So here, we see 5 layers of different types of neurons. The photoreceptors, which includes the rods and cones, horizontal cells, bipolar cells, amacrine cells, and the retinal ganglion cells. The photoreceptors, which are referred to as rods and cones, are sensory neurons that detect light. They are the transducers of the visual system. They convert light into electrical signals that travel through other retinal neurons to reach the optic nerve. Bipolar cells. Bipolar cells are responsible for transmitting signals from photoreceptors to a retinal ganglion cell, whose axon form the optic nerve which carries information to the brain. Meanwhile, there's also a lateral communication that happens in the retina. The horizontal cells aid in communication between photoreceptors and bipolar cells. They regulate the signal that emerges from several rods and cones. There's also amacrine cells that aid communication between bipolar and retinal ganglion cells. And lastly is the retinal ganglion cell. It relays signals from bipolar and amacrine cells to the brain through their axons that form the optic nerve. The retina is actually built in an inside-out arrangement, meaning light reaches the receptor layer only after passing through other 4 layers. Then, once the receptors have been activated, the neural message is transmitted back out through the retinal layers to the retinal ganglion cells, whose axons project across outside of the retina. So the idea that we get here is that there are a lot of cells involved. Several communications is made in order for us to perceive the world. to see colors and the details of our surroundings. It's just so amazing, right? However, the inside out arrangement of the retina actually creates two visual problems. One is that incoming light is distorted by the retinal tissue through which it must pass before reaching the receptors. The other is that for the bundle of retinal ganglion cells, axons, to leave the eye, there must be a gap in the receptor layer. This gap is called the blind spot. Blind spot is the area on the retina without receptors that respond to light. Therefore, an image that falls on this region will not be seen. It is also in this region that the optic nerve exits the eye on its way to the brain. And how is it solved? Blind spot is solved by our own visual system as it uses information provided by the cells around the blind spots to fill in the gaps in your retinal images. This process is what we call completion. Aside from responding to blind spot, through completion process, the visual system extracts key information about the object, primarily information about its edges and their location, and conducts that information to the cortex where a perception of the object is created from that partial information. For example, the color and brightness of a large unpatterned surfaces are not perceived directly, but are filled in or completed by a completion process called surface interpolation. This is a process by which we perceive surfaces. The visual system extracts information about the edges and from it infers the appearance of large surfaces. Meanwhile, the first of these two problems is minimized by the fovea. Fovea is an indentation at the center of the retina that is specialized for high acuity vision or for seeing fine details. So notice in this picture that there is the thinning of the retinal ganglion cell layer which reduces the distortion of incoming light. Another is that fovea only consists of cones, which gives us very high acuity and four-color detailed information. So, if you want to place your best sight on an object, you should put it right in front of your eyes because that's going to pick up on the fovea. Now, let's take a closer look on the two different types of photoreceptors in the human retina, the cones and the rods. Looking at their shapes, the origin of their names becomes quite obvious. The existence of these two was first noticed that species active only in the day tend to have cone-only retinas, and species active only at night tend to have rod-only retinas. From this observation emerged the duplexity theory of vision, the theory that cones and rods mediate different kinds of vision. Cones are what we refer to as the photopic system. They are used for daytime vision and very high acuity and rich in color. On the other hand is rods, or what we call the scotopic system. This is used for nighttime vision and is high sensitivity, allowing for low acuity vision in dim light, yet produces a kind of perception that lacks details and color information. The differences between scotopic and photopic vision result in part from a difference in the way the two systems are wired. As you can see here, there's much more convergence in the rod system or in the rod-fed circuits. You see how there's 8 rods where each one is converging onto a bipolar cell and onto a retinal ganglion cell. This increases the sensitivity, all the while decreasing the acuity. On the other hand, low convergence is seen in cone-fed circuits. Here, one cone influences a bipolar cell and that bipolar cell influences a retinal ganglion cell, all the while decreasing the acuity. On the other hand, low convergence is seen in cone-fed circuits. Here, one cone influences a bipolar cell, and that bipolar cell influences a retinal ganglion cell. So less convergence here, resulting to less sensitivity but high acuity. Cones and rods also differ in their distribution on the retina. There are no rods at all in the fovea, only cones. Then, moving out from fovea, there are fewer cones and many rods. The sensitivity towards particular wavelengths of light is slightly different for these two receptor cells as well. We can see it here in the spectral sensitivity curve, which is a graph that shows the relative brightness of lights of the same intensity presented at different wavelengths. Humans and other animals with both cones and rods have two of them, a photopic spectral sensitivity curve and a scotopic spectral sensitivity curve. With photopic vision, of course mediated by the cones, the visual system is maximally sensitive to wavelengths of about 560 nanometers, which is roughly yellow light as you can see here in the graph. Thus, under photopic conditions, a light at 500 nanometers, which is sort of a bluish to greenish light, would have to be much more intense than one at 560 nanometers to be seen as equally bright. That's for photopic vision. In contrast, scotopic vision mediated by the rods, maximum sensitivity happens around 500 nanometers, sort of a bluish green light. Thus, under scotopic condition, a light of 560 nanometers, this time the yellow light, would have to be much more intense or brighter than one at 500 nanometers to be seen as equally bright. As light dims and our eyes switch from photopic vision to scotopic vision, an interesting visual effect can be observed that relates to this discrepancy in photopic and scotopic spectral sensitivity, and it is called the Purkinje Effect. Purkinje Effect, named after the man who first discovered and observed this phenomenon, John Purkinje, is the tendency for the peak luminance sensitivity of the human eye to shift towards the blue end of the color spectrum at low illumination levels. Because, as we know, when light is abundant or if it is in daytime, yellow and red objects will appear brighter than green and bluer ones. Yet, as the light dims, this relative brightness will appear to reverse. Look around you. Wait, just look at the screen. What you see is not a few color details at the center of a grayish scene. What you see is an expansive, richly detailed, and lavishly colored world. But if cones are responsible for mediating high acuity color vision under photopic conditions, like in this video, how do you think they accomplish their task when most of them are crammed into the fovea? Although we are not aware of it, the eyes continuously scan the visual field, and our visual perception at any instant is a summation of recent visual information. It is because of this temporal integration that the world does not vanish momentarily each time we blink. Our eyes continuously move even when we try to keep them fixated. The involuntary fixational eye movements are of three kinds: the tremor, drifts, and saccades. We will just give emphasis on saccades. Saccades refer to fast conjugate eye movements that shift the eyes from one target to another, bringing an object of interest into focus on the fovea, where visual acuity is highest. So when eye movements or their main effect are blocked, visual objects begin to fade and disappear. This happens because most visual neurons respond only to changing images. If retinal images are artificially stabilized, the images start to disappear and reappear. Thus, eye movements enable us to see during fixation by keeping the images moving on the retina. To put it simply, the principal function of eye movements is to enhance visual acuity by keeping images stabilized on the retina to reduce blur. So now we have a basic grasp of how light is received by the retina. But how is this converted into information for the brain? Visual transduction. Visual transduction is the conversion of light to neural signals by the visual receptors. A breakthrough in the study of visual transduction came in 1876, when a red pigment called rhodopsin was extracted from rods. When the rhodopsin was exposed to continuous intense light, it was bleached or lost its color, and also lost its ability to absorb light. But when it was returned to the dark, it regained both its redness and its light absorbing capacity. These actually make sense, since from here we can infer that rhodopsin's absorption of light is the first step in rod-mediated vision. That's why it is shown here that the degree to which rhodopsin absorbs lights of different wavelengths is related to the ability of humans and other animals to detect the presence of different wavelengths of light under scotopic conditions. Rhodopsin is a G protein coupled receptor that responds to light rather than to neurotransmitter molecules. Unlike any other G protein coupled receptors, it initiates a cascade of intracellular chemical events when they are activated. When rods are in darkness, their sodium channels are partially open, thus keeping the rods slightly depolarized, generating a graded potential which results in a steady flow of glutamate, which is an excitatory neurotransmitter. However, when rhodopsin receptors are bleached by light, the sodium channels close, hyperpolarizing the rods and reduces or inhibits the release of glutamate. The transduction of light by rods exemplifies an important point that signals are often transmitted through neural systems by inhibition. Guys, I hope you're still there. So one reporter is done, and before we proceed to the next, we'll have a quick icebreaker entitled Finding Psychmates. Since we're all about the coordination of the eyes and brain right now, we want to test how sharp your eyes and how active is your brain in the present. What you're going to do is just simple. You guys just have to spot our classmate in the picture that we'll show to you, then answer the question he or she will give to you. So let's go!
Many pathways in the brain carry visual information, and by far, the largest and most thoroughly studied visual pathway are the retina geniculate striate system. Just a few facts about retina geniculate striate system. So, about 90% of axons of retinal ganglion cells become part of retina geniculate striate. No other sensory system has such a predominant pair of pathways to the cortex. And retina geniculate striate system conduct signals from each retina to the primary visual cortex or striate cortex via the lateral geniculate nuclei of the thalamus. So I know you're wondering, what is primary visual cortex? What is lateral geniculate nuclei? Let's dig. The organization of the visual pathways. So the main idea to take away from this figure is that all signals from the left visual field reach the primary visual cortex either ipsilaterally from the temporal hemiretina of the eye or contralaterally via the optic chiasm from the nasal hemiretina of the left eye, and that to make it simpler, these two lateral geniculate nuclei, so each one receives information from both eyes, and we have these two eyeballs. The information from the temporal hemiretina, ito, stays ipsilateral or on the same side of the body, and the nasal hemiretina crosses over sa optic chiasm. O yung temporal hemiretina, kung nagsistay lang siya ipsilateral or on the same side of the body, while the nasal hemiretina siya yung nagko-cross over. So LGN have information from both eyes, but only from the left side of the world or from the right side of the world. Nasal hemiretina and temporal hemiretina. So primates have a partial decussation pattern whereby only the optic nerve fibers from the nasal hemiretina cross the contralateral side, while those of the temporal hemiretina project ipsilateral. So yun na nga yung sinabi ko kanina. Yung temporal, dito lang siya sa gilid, sa side niya. Pero yung nasal hemiretina, nagko-crossover siya sa optic chiasm. Optic nerve. It is the second of several pairs of cranial nerves. It transmits sensory information for vision in the form of electrical impulses from the eye to the brain. So, ito yung optic nerve. Ito siya. Ganyan yung pagta-travel ng sensory information. So, optic chiasm. Optic chiasm is located in the forebrain, directly in front of the hypothalamus. So, the left and the right optic nerves intersect at the chiasm, thus creating the hallmark X shape. So, yung optic nerve dito siya nag-intersect sa optic chiasm. Yung nasal hemiretina, dito siya nag-intersect. And optic tract. The optic tract primarily conveys visual information from the optic chiasm to the lateral geniculate nuclei. So, yung optic nerve na nag-intersect dito sa optic chiasm, dito siya napupunta sa optic tract, and then yun yung nagko-convey ng information papunta sa lateral geniculate nuclei. Primary visual cortex. The visual cortex is the primary cortical region of the brain that receives, integrates, and processes visual information relayed from the retina. It is often referred as the striate cortex. So, dito siya nakalocate, and yung mga information na nag-gather, dito na siya pinaprocess sa primary visual cortex. Lateral geniculate nuclei, a multi-layered structure that receives input from both eyes to build a representation of the contralateral visual hemifield. So, yung kanina, yung nandun sa optic tract, mapupunta na siya dito sa lateral geniculate nuclei. It is multi-layered because each of the lateral geniculate nuclei has 6 layers, and each layer of nucleus receives input from all parts of the contralateral visual field of one eye. Ito yung layers ng lateral geniculate nucleus. Retinotopic organization. Retinotopy, also known as retinal mapping, is the mapping of visual information from the retina to neurons in the brain. So the parts of the brain which receives visual information are organized into retinotopic map. So yung ano natin, yung retina geniculate stride system is retinotopic because each level of the system is organized like a map of the retina. Retinotopic organization. This is a demonstration of the retinotopic organization of a primary visual cortex that was provided by Dobel, Miladejovsky, and Gervin in the 1974. So they implanted an array of electrodes in the primary visual cortex of patients who were blind because of damage to their eyes. Yan. Naglagay sila ng uh, micro ng electrodes dyan. If electrical current was administered simultaneously through an array of electrodes forming a shape such as a cross on the surface of the patient's cortex, the patient reported seeing a glowing image of that shape. So this finding and related work on retinal implants could be the basis for the development of visual processes that could benefit many blind people. M channels and P channels. Parallel channel of communication flows through each lateral geniculate nucleus. So P channel or parvocellular layer, paro means small, so parvocellular layer are composed of neurons with small cell bodies, and they are particularly responsive to colors, fine pattern details, and stationary or slowly moving objects. And cones provide the majority input of the P layers. M channel or magnocellular layer. Magno means large. So magnocellular layer are composed of neurons with large cell bodies. The magnocellular neurons are particularly responsive to movement, and rods provide the majority input of the M layer. One channel runs through the top four layers, and this layer are called the parvocellular layer. So dito yung flow niya. Siya dito yung nagra-run ng ano dito. And the other channel runs through the bottom two layers. Dito naman yung magnocellular layer. Dito din. So, if nagtataka kayo kung ano yung CI, that is contralateral, ipsilateral. So, one of the layer in this in the lateral geniculate nucleus is contralateral, or yung information na galing dito sa right eye, dito siya napupunta sa P channel, sa dito sa P channel na to. Ganon din sa ipsilateral, sa magno, ganyan. Same lang yung nangyayari dito. Seeing edges. The perception of an edge is really the perception of a contrast between two adjacent areas of the visual field. The non-existent stripes of brightness and darkness running adjacent to the edges, they enhance the contrast at each edge and make the edge easier to see. That is what we call the match. Next na ako eh. Receptive field of visual neurons. The Nobel Prize winning research of David Hubble and Thorsten Wiesel is the fitting focus of this discussion because their research talks about brightness contrast. Hubble and Wiesel's method is a technique for studying single neurons in visual system. And it their research has revealed much about the neural mechanisms of vision, and subsequent generation of sensory neurophysiologists have adapted their method. Their method is a technique for studying single neurons in the visual system of laboratory animals. So their research subjects were cats and monkeys. So this is the procedure and how they did their research. Now let's look into the procedure of the research and the Wiesel's research. So first, the tip of a microelectrode is positioned near a single neuron in the part of visual system under investigation. So during testing, eye movements are blocked by paralyzing the eye muscles, and the images on the screen in front of the subject are focused sharply on the retina by an adjustable lens. Next step in the procedure is to identify the receptive field of the neuron. But what is the receptive field? Uh, Receptive field of a visual neuron is the area of the visual field within which it is possible for a visual stimulus to influence the firing of that neuron. The final step in the method is to record the responses of the neuron to various simple stimuli within its receptive field in order to characterize the types of stimuli that most influence its activity. So, um, then the electrode is advanced slightly, and the entire process of identifying and characterizing the receptive field properties is repeated for another neuron, and then for another, and so on. The general strategy is to begin by studying neurons near the receptors and gradually work up through higher and higher levels of the system in an effort to understand the increasing complexity of the neural responses at each level. Match bands. So here here we have two rectangles. One is darker and the other one is lighter. Here it illustrates how visual processing can lead to something interesting things, and to a certain extent it shows that what you see is not always what's there. So I'm gonna put them together, this solid gray, lighter solid gray, and darker solid gray. I'm gonna put them together and notice that here at the border you're gonna see that here is just a little lighter than the other here. So it's amazing na you can't really see something here but if you pero pag pinaghalo or pinagdikit mo siya, makikita mo yung match bands. Yan yung match band dito. But there really isn't lighter edge there, but there's something happening inside our eyes that making it seems as if it's lighter. Diba? Parang lighter siya dito, pero they're both gray and dark. Pero mas darker na naman to, pero parang dito lang kung ano nangyayari dito. Ganon. That's the match bands. Improve the perceptibility of objects in the scene by enhancing the brightness difference between objects and their backgrounds is called contrast enhancement. So, although we are normally unaware of it, every edge we look at is highlighted for us by the contrast enhancing mechanisms of our nervous system. So, in effect, our perception of edges is better than the real thing, as determined by measurements of the physical properties of the light entering our eyes. So, the classic studies of the physiological basis of contrast enhancement were conducted on the eyes of horseshoe crab. The lateral eyes of a horseshoe crab are ideal for certain types of neurophysiological research. So, unlike mammalian eyes, they were composed of very large receptors called ommatidia, each with its very own large axon. The axons of the ommatidia are interconnected by a lateral neural network. So, in order for us to understand the physiological basis of contrast enhancement in the horseshoe crab, we must know two things. First, the first is that if a single ommatidium is illuminated, it fires at a rate that is proportional to the intensity of the light striking it. So, more intense lights produce more firing. The second is that when the receptor fires, it inhibits its neighbors via lateral neural network. This inhibition is called lateral inhibition because it spreads laterally across the array of receptors. So, the amount of lateral inhibition produced by a receptor is greatest when the receptor is most intensely illuminated, and the inhibition has its greatest effect on the receptor's immediate neighbors.
Finding segments!
Good day, everyone. Continuing the report of my groupmate, I will be discussing the neurons of the retina geniculate striate system. Hubble and Weasel began their studies of visual system neurons by recording from the three levels of the retina geniculate striate system. First, from retinal ganglion cells, then from lateral geniculate neurons, and finally, from the striate neurons of lower layer 4. They found little change in the receptive fields as they worked through the levels. When Hubble and Weasel compared the receptive fields recorded from the retinal ganglion cells, lateral geniculate nuclei, and lower layer 4 neurons, commonalities were readily apparent. So, these are first, at each level, the receptive fields in the foveal area of the retina were smaller than those at the periphery. This is consistent with the fact that fovea mediates fine-grained division. Second, all the neurons had receptive fields that were circular. Third, all the neurons were mononuclear, that is, each neuron had a receptive field in one eye but not the other. And lastly, many neurons at each of the three levels of the retina geniculate striate system had receptive fields that comprised an excitatory area and an inhibitory area separated by a circular boundary. Continuing with their experiment, when Hubble and Weasel shone a spot of achromatic light onto the various parts of the receptive fields, of a neuron in the retina geniculate striate pathway, they discovered two different responses. The neuron responded with either on-firing or off-firing, depending on the location of the spot of light in the receptive field. That is, the neuron either displayed a burst of firing when the light was turned on, or displayed an inhibition of firing in the burst of firing when it was turned off. So, for most of the neurons in the retina geniculate striate system, the reaction on firing or off firing to a light in a particular part of receptive field was quite predictable. It depended on whether they were on center cells or off center cells, as you can see on a picture on the left. So, on-center cells respond to lights shown in the central region of the receptive fields with on-firing and to lights shown in the periphery of the receptive fields with inhibition, followed by off-firing when the light is turned off, while off-center cells is the opposite pattern. They respond with inhibition and off-firing in response to light in the center of the receptive fields with on-firing to lights in the periphery of the receptive fields. So, in effect, on-center and off-center cells responds best to contrast. The picture in the right illustrates this point. The most effective way to influence the firing rate of an on-center or off-center cell is to maximize the contrast between the center and the periphery of its receptive field by illuminating either the entire center or the entire surrounding, while leaving the other region completely dark. So now we move on to simple and cortical cells, or complex cells. The receptive fields of most primary visual cortex neurons fall into one of two classes, simple or complex. Neither of these classes includes the neurons of the lower layer 4. So, the first one is the simple cells. They are like lower 4 neurons. They have receptive fields that can be divided into antagonistic on and off regions and are thus unresponsive to diffuse light. And like lower 4 neurons, they are monocular. The main difference is that the borders between the on and off regions of the cortical receptive fields of simple cells are straight lines rather than circles. So, complex cells are most numerous than simple cells. Like simple cells, complex cells have rectangular receptive fields and respond best to straight line stimuli in a specific orientation and are unresponsive to diffuse of light. However, complex cells differ from simple cells in three important ways. First, they have larger receptive fields. Second, it is not possible to divide the receptive fields of complex cells into static on and off regions. It responds to a particular orientation regardless of its position within the receptive field. So, meaning that a cell will respond continuously to as it moves across the field. And third, unlike simple cortical cells, which are all monocular, many complex cells are binocular. So, most of the binocular cells in the primary visual cortex of monkeys display some degree of ocular dominance. That is, they respond more robustly to stimulation of one eye than they do to the same stimulation to the other. And in addition, some binocular cells fire best when preferred stimulus is presented to both eyes at the same time, but in slightly different positions on the two retinas. In other words, these cells respond best to retinal disparity and thus are likely to play a role in depth perception. So here is another side-by-side comparison about simple and complex cells, as you can see here. Moving on. After describing the receptive fields of visual cortex neurons, Hubble and Wiesel focused their analysis on how neurons with different receptive fields are organized in primary visual cortex. So they reached three important conclusions about the organization of primate visual cortex. So first, they concluded that the primary visual cortex was organized into functional vertical columns. All of the neurons in the same vertical column respond to stimuli applied to the same area of the retina, and they are dominated by the same eye and prefer the same straight line angles. Second, they found that the location of various functional columns in primary visual cortex is influenced by the location on the retina of the column's visual fields, by the dominant eye of the column, and by dominant column's preferred straight line angle. Hubble and Wiesel concluded that all the functional columns in the primary visual cortex that analyzes input from one area of the retina and are clustered together, and that half of a cluster receives input from the left eye and the other half receives input from the right eye, and that each cluster includes neurons with preferences for straight line stimuli of various orientations. And third, as Hubble and Wiesel studies progressed from retina to thalamus to lower layer 4 of visual cortex and to simple cortical cells to complex cortical cells, the preferences of the neurons became more complex. Hubble and Wiesel concluded that this occurred because neurons with simpler preferences converged on neurons with more complex preferences. Most investigations of the responsiveness of visual system neurons are based on two implicit assumptions. The first is that the mechanisms of visual processing can be best identified by studies using simplified, controllable, artificial stimuli. The second is that the receptive field properties of each neuron are static, unchanging properties of that neuron. So, studies of the responses of visual cortex to natural scenes, just the type of scenes the visual system has evolved to perceive, indicate that the response of a visual cortex neuron depends not only on the stimuli in its receptive field, but on the larger scene of which these stimuli are embedded. The influences on a visual neuron's activity that are caused by stimuli outside the neuron's receptive field are generally referred to as contextual influences. So, contextual influences can take many forms depending on the exact timing, location, and the shape of the visual stimuli under investigation and on the ambient light levels. So now let's talk about seeing color and the component and deponent processing. So, color is one of the most obvious qualities of human visual experience. Black is experienced when there is an absence of light, while the perception of white is produced by an intense mixture of a wide range of wavelengths in roughly equal proportions. And the perception of gray is produced with the same mixture at lower intensities. And now, we will deal with the perception of colors such as blue, green, and yellow. To start off, we have the component theory or the trichromatic theory of color vision. So, it was proposed by Thomas Young in 1802 and refined by Hermann von Helmholtz in 1852. So, according to this theory, there are three different kinds of color receptors, each with a different spectral sensitivity, and the color of a particular stimulus is presumed to be encoded by the ratio of activity in the three kinds of receptors. Young and Helmholtz derived their theory from the observation that any color of the visible spectrum can be matched by a mixing together of three different wavelengths of light in different proportions. So, we also have this opponent process theory of color vision. It was proposed by Ewald Hering in 1878. He suggested that there are two different classes of cells in the visual system for encoding color and another class for encoding brightness. Do you think that he was right? And yes, we will see. So, Hering hypothesized that each of the three classes of cells encoded two complementary color perceptions. One class of color coding cells signaled red by changing its activity in one direction, such as hyperpolarization, and signaled red's complementary color, green, by changing its activity in the other direction, such as depolarization. And another class of color-coding cells was hypothesized to signal blue in its complement yellow. In the same opponent fashion, in the class of brightness-coding cells was hypothesized to similarly signal both black and white. So we have here an example of a color after image that is related to the opponent process theory. So stare at the white dot in the center of this oddly colored flag for about 30 seconds. You may pause this video to do this. After 30 seconds, look at a white piece of paper or white wall. Notice that the colors are now the normal, expected colors of the American flag. They are also the primary colors that are the opposites of the colors in the picture. And most importantly, the evidence for the opponent process theory of color vision. So have you already seen it? Now we move on to complementary colors. Complementary colors are a pair of colors, for example, green light and red light, that produce white or gray when combined in equal measure. Herring based his opponent process theory of color vision on several behavior observations. One was that complementary colors cannot exist together. He said, there is no such thing as bluish yellow or reddish green. Another one was that the after image produced by staring at a red is green and vice versa, and the after image produced by staring at yellow is blue and vice versa. So an example of this is by doing this activity. Have you ever noticed complementary after images? To see them, stare at the fixation point in the left panel for 1 minute without moving your eyes. The X in the middle of the colors. Then, quickly shift your gaze to the fixation point in the right panel. In the right panel, you will see 4 squares whose colors are complementary to those in the left panel. Neither component nor opponent processing can account for the single most important characteristic of color vision, which is the color constancy. So, color constancy refers to the fact that the perceived color of an object is not a simple function of the wavelengths reflected by it. So, color constancy is the tendency for an object to stay the same color despite major changes in the wavelengths of light that it reflects. Although color constancy is an important feature of our vision, we are normally unaware of it. Under everyday conditions, we have no way of appreciating just how much the wavelength reflected by an object can change without the object changing its color. So, moving on, according to Land's Retinex's theory of color vision, the color of an object is determined by its reflectance, the proportion of light of different wavelengths that a surface reflects. So, he said in his theory, that relative wavelengths are constant, so perception is constant. It's very important for neuroscientists who are trying to discover the neural mechanisms of color vision. Why? Because it suggests how some of the neurons involved in color vision must work. If the perception of color depends on the analysis of contrast between adjacent areas of the visual field, then some color neurons must be responsive to color contrast. And they are. For example, dual opponent color cells. Dual opponent color cells are sensitive to color contrast, and during their time, they tried to study it on monkeys. To the relief of instructors and students alike, the term blobs has become the accepted scientific label for peg-like cytochrome oxidase rich dual opponent color columns. And lastly, functional MRI studies have provided evidence of dual opponent color cells in the human visual cortex.
Finding segments!
Good day, everyone. I am Andromeda de la Cruz. I am going to be your reporter for this final part of Chapter 6, and we are going to discuss the cortical mechanisms of vision and conscious awareness. So basically, this is more of the entire occipital cortex. So without further ado, let's start. So now let's begin, okay, our formal discussion. We have three different classes of visual cortex. So the visual cortex is often considered to be of three different classes. One, we have the primary visual cortex. This is the area of the cortex that receives most of its input from visual relay nuclei of the thalamus. The primary visual cortex is located in the posterior region of the occipital lobes. And second, we have the secondary visual cortex. Those that receive input from the primary visual cortex. So, yung mga nakukuha na input ni secondary visual cortex nanggagaling kay primary visual cortex. Most areas of the secondary visual cortex are located in two general regions. One in the prestriate cortex, over here, as you can see in the image, the prestriate cortex, and in the inferotemporal cortex. These two areas here. The prestriate cortex is the band of tissue in the occipital lobe that surrounds the primary visual cortex, and the inferotemporal cortex, this one, is the cortex of the inferior temporal lobe. Okay? And lastly, we have the visual association cortex. So this cortex are those that receive input from areas of secondary visual cortex. So yung mga nakukuha niya, the input that the visual association cortex receives is from the secondary visual cortex. Areas of association cortex that receive visual input are located in several parts of the cerebral cortex, but the largest single area is in the posterior parietal cortex, which is located over here. Okay, so those are the three different classes of visual cortex. Scotoma is an area of blindness in the corresponding area of the contralateral visual fields, or contralateral meaning opposite sides of the visual field of both eyes due to damage in the primary visual cortex. Okay, so patients with scotomas are usually given perimetry tests with suspected damage in the primary visual cortex. And so in this image here, we have this patient or subject with a bullet wound in his left primary visual cortex. So the scotomas, or the areas of blindness, are indicated here. These are his scotomas. So yung mga scotoma niya nasa right visual fields ng both eyes niya. Okay? This is the result of his perimetry test. And so how does a perimetry test work? So while the patient's head is held motionless on a rest chin, just like here in this image, the patient stares with one eye at the fixation point on a screen. A small dot of light is then flashed on various parts of the screen, and the patient presses a button, this one here, he's holding a button, to record when the dot is seen. He presses a button to record when the dot is seen. And then the entire process is repeated for the other eye. So this test is only done one eye at a time. The result is a map of the right and left visual field of each eye, which indicates any areas of blindness. Ayan. So this is a more accurate depiction of the result of a perimetry test. On the left is a normal visual field. The black spot on the left side is the normal blind spot, di ba kanina, just like our quick illusion. And on the right is an abnormal visual field showing defects that involve both central and peripheral vision. These visual defects are likely to be noticed by the patient. So many patients with scotomas are not consciously aware of their deficits, and this is because of completion. So scotoma patients who look at complex figures in parts of their scotoma often report seeing a complete image, and completion may depend on residual visual capacities in the scotoma. So hemianopsic patients, or patients having a scotoma covering half of the visual field, may see an entire face when they focus on a person's nose or on a certain point, even when the side of the face in the scotoma has been covered by a blank card. So now let's move on to conscious awareness. So you probably equate perception with conscious awareness. That is, that you assume that if a person sees something, he or she will be consciously aware of seeing it, di ba? So in everyday thinking, perceiving and being aware are inseparable processes. We assume that someone who has seen something will be able to acknowledge that he or she has seen it and be able to describe it. But there are phenomena for which this is not the case. There are people who see things but have no conscious awareness of them. And this is where blind sight comes in. So blind sight is the ability to respond to visual stimuli in a scotoma with no conscious awareness of them. So of all visual abilities, perception of motion is most likely to survive damage through primary visual cortex. So a patient with blind sight might reach out and grab a moving object in her scotoma, all the while claiming not to see the object. Blind sight is sometimes displayed by patients with scotomas resulting from damage to primary visual cortex. No? So if blind sight confuses you, imagine how it confuses people who experience it. Okay, so this is kind of confusing. So let's move on to one case regarding blind sight. So the case of DB, the man confused by his own blind sight. So DB had no awareness of seeing in his blind left field. Despite the blindness, he could accurately reach for visual stimuli in his left field and could accurately differentiate between a horizontal and the diagonal line in his left field if forced to guess. Diba? On the previous slide, we could see, diba, there was an image of a man that was forced to guess kung ano daw yung nakikita niya. Even though the patient claimed that he could not see anything, but if he was forced to guess, he was accurately able to describe what he saw. So he was, ayan, naka-describe siya ng horizontal and diagonal line if he was forced to guess, or DB. So why is this so? So here are the neurological interpretations of blind sight. So one, the striate cortex daw is not completely destroyed, and the remaining islands of functional cells are capable of mediating some visual abilities in the absence of conscious awareness. And second, the visual pathways that ascend directly to the secondary visual cortex from the subcortical visual structures without passing through the primary visual cortex are capable of maintaining some visual abilities in the absence of cognitive awareness. Diba kanina we discussed the three classes of the visual cortex? We have the primary visual cortex, the secondary visual cortex, and the visual association cortex, and that the major flow of information is from primary to secondary to association. So it says here that the secondary visual cortex from the subcortical visual structures without passing through the primary visual cortex are capable of maintaining some visual abilities in the absence of cognitive awareness. So there are some supports for both theories, but it is far from conclusive in either case. Secondary visual cortex and the portions of association cortex involved in visual analysis are both composed of many different areas, and each specialized for a particular type of visual analysis. So now let's move on to the functional areas of the secondary and association visual cortex. So we're done with the primary visual cortex. Okay, so neurons in each functional area in the secondary and association visual cortex respond most vigorously to different aspects of visual stimuli. For example, to color, to movement, to shape. So parang the characteristics or aspects of visual stimuli. So it has been found that there are more than 30 different functional areas of the primary visual cortex, and there are 24 areas of the secondary visual cortex and 7 areas of association visual cortex. So researchers have so far delineated about a dozen different functional areas of human visual cortex. Okay? So here on the picture, makikita natin yung, it's not, we can't tell, but these are the different areas of the primary visual cortex, the secondary visual cortex, and the association visual cortex. So may mga individual labels sila. This is MTV5, VF4. So each of them respond vigorously to different aspects of visual stimuli. So MTV5 may probably respond to motion, siguro, sabihin na lang natin, something like that. So, may kanya-kanya silang mga characteristics. They respond to different characteristics sa visual stimuli. So, selective lesions to the different areas produce different visual losses, and there are anatomical and organizational differences among the areas. Anterograde and retrograde tracing studies have identified more than 300 interconnecting pathways. So connections between areas are virtually always reciprocal. So parang two-way sila. It's not just one way. They are all interconnected with each other. So PET, fMRI, and evoked potentials have been used to identify various areas of visual cortex in humans. So they gathered different volunteers whose brains have been monitored while they inspect various types of visual stimuli. So by identifying the areas of activation associated with various visual properties, a map of some of these areas are shown. In most are similar in terms of location and anatomical characteristics and function to areas already identified in a previous study. So, kumuha sila ng mga volunteers where they monitor yung mga brain while they were showing these volunteers different visual stimuli. For example, a red car, a long hammer, parang ganon. And then they noticed that different parts of the visual cortex parang nag-fire up whenever some specific characteristics of the visual stimuli were being shown to the volunteers. Now we have the dorsal and ventral streams. So what are these streams? So pathways that conduct information from the primary visual cortex through various specialized areas of secondary and association cortex can be thought of as components of two major streams, the dorsal stream and the ventral stream. So the dorsal stream flows from the primary visual cortex to the dorsal pre-striate cortex to the posterior parietal cortex. Okay, so visual cortex neurons respond robustly to spatial stimuli. So the neurons in the dorsal stream, again, respond robustly to spatial stimuli. Okay, this is important to take note of. While on the ventral stream flows from the primary visual cortex to the ventral pre-striate cortex to the inferotemporal cortex. And for the ventral stream, the neurons here respond to the characteristics of objects, such as the color and shape. Okay, so in this figure, we can see the two streams. So this one here, we have the dorsal stream. The dorsal stream flows from the primary visual cortex to the dorsal pre-striate cortex to the posterior parietal cortex. And on the other hand, we have the ventral stream, this one here. So the ventral stream flows from the primary visual cortex to the ventral pre-striate cortex to the inferotemporal cortex. So as I mentioned kanina, most visual cortex neurons in the dorsal stream, this here, respond most robustly to spatial stimuli, such as those indicating the location of objects or their direction of movement. And there are clusters of visual neurons in the ventral stream, over here, and each cluster responds specifically to a particular class of objects. For example, most neurons in a particular cluster may respond to faces, whereas most neurons in another cluster might respond to animals. So on the ventral stream, more on the visual characteristics, while on the dorsal stream, more on the spatial stimuli of an object or visual stimuli. So now let's move on to the two theories that try to discuss the primary functions of the dorsal stream and the ventral stream. So first we have the where versus what theory. This theory is by Ungerleider and Mishkin, and they propose that the dorsal and ventral visual streams perform different visual functions. So according to this theory, the dorsal stream is involved in the perception of where objects are located, while the ventral stream is involved in the perception of what objects are. So the major implication of the where versus what theory is that damage to some areas of cortex may abolish certain aspects of vision while leaving others unaffected. The most convincing support for the influential where versus what theory has come from the comparison of the specific effects of damage to the dorsal and ventral streams. So, patients with damage to the posterior parietal cortex often have difficulty reaching accurately for objects they have no difficulty describing. So, ayan, patients with damage to the posterior parietal cortex, diba? As we discussed, this is where the dorsal stream is located. Yes, located. And conversely, patients with damage to the inferotemporal cortex, or where the ventral stream runs down or is located, often have no difficulty reaching accurately for objects they have difficulty describing. So let's read it this way na lang. Patients with damage to the dorsal stream or the posterior parietal cortex often have difficulty reaching accurately for objects. Why so? Because it is said in this theory that the dorsal stream is involved in the perception of where objects are located. So the patients with damaged dorsal streams have difficulty reaching accurately for objects, but they have no difficulty describing objects. Conversely, patients with damage to the ventral stream or the inferotemporal cortex often have no difficulty reaching accurately for objects, but have difficulty describing them. Because, again, the ventral stream is involved in the perception of what objects are, according to this theory. So for the second theory, we have the control of behavior versus conscious perception theory. So this is by Goodale and Milner. So they argued that the primary difference between the dorsal and ventral streams is not the kinds of information they carry, but the use to which that information is put. So they argued that the dorsal stream is to direct behavioral interactions with objects and not just simply where the objects are, or not just simply about spatial stimuli, but direct behavioral interactions with objects, while the ventral stream is to mediate the conscious perception of objects, and in contrast with the previous theory, not just what or not just what the characteristics of of an object is or are. So the control of behavior versus conscious perception theory can actually readily explain the two major neuropsychological findings that are the foundation of the where versus what theory. So namely, the control of behavior versus conscious perception theory suggests that patients with dorsal stream damage may do poorly on tests of location and movement because most tests of location and movement involve performance issues, and that patients with ventral stream damage may do poorly on tests of visual recognition because most tests of visual recognition involve verbal responses and thus conscious awareness. So in contrast with the so the two theories, okay? So the where versus what theory suggests that the dorsal stream is involved in the perception of where objects are located, but in the control of behavior versus conscious perception theory, the dorsal stream now is to direct behavioral interactions with objects. So it is said here that patients with dorsal stream damage may do poorly on tests of location and movement because most tests of location and movement involve performance measures. So this statement here is more inclined or is most closely related to the control of behavior versus conscious perception theory because it says that the dorsal stream is to direct behavioral interactions with objects and therefore it involves performance measures. And the second one, patients with ventral stream damage may do poorly on tests of visual recognition because most tests of visual recognition involve verbal responses. So meaning, the ventral stream is more likely to mediate the conscious perception of objects and not just the simple perception of what objects are. Okay, so the major support for the control of behavior versus conscious perception theory is the confirmation of its two primary predictions. One, we have that some patients with bilateral lesions to the ventral stream may have no conscious experience of seeing and yet be able to interact with objects under visual guidance. And number two, some patients with bilateral lesions to the dorsal stream may consciously see objects but be unable to interact with them under visual guidance. So we have this image here to sum up the two theories, the dorsal and ventral streams, and the two theories and what they predict. So again, for the where versus what theory, it says that the dorsal stream specializes in visual spatial perception, while the ventral stream specializes in visual pattern recognition. This theory also predicts that damage to the dorsal stream disrupts visual spatial perception, while damage to the ventral stream disrupts visual pattern recognition. So the other one is the control of behavior versus conscious perception theory. So in this theory, it says that the dorsal stream specializes in visually guided behavior, while the ventral stream specializes in conscious visual perception. This also predicts that damage to dorsal stream disrupts visually guided behavior but not conscious visual perception. And damage to ventral stream disrupts conscious visual perception but not visually guided behavior. So much for Dora. Now let's move on to an actual case of damage to the two streams, to the dorsal stream and to the ventral stream. Okay, so the first one, we have the case of DF, the woman who could grasp objects she did not consciously see. So DF has a bilateral damage in the ventral prestriate cortex, so thus interrupting the flow of the ventral stream. Amazingly, she can respond accurately to visual stimuli that she does not consciously see. So despite her inability to consciously recognize the size, shape, and orientation of visual objects, DF displayed accurate hand movements directed at the same objects. So, for example, when she was asked to indicate the width of blocks with her index finger and thumb, her matches were variable and unrelated to the actual size of the blocks. However, when she was asked to pick up blocks of different sizes, the distance between her index finger and thumb changed appropriately with the size of the objects. So in other words, DF adjusted her hand to the size of the objects she was about to pick up, even though she did not consciously perceive their size. So a similar dissociation occurred in her responses to the orientation of stimuli. When presented with a large slanted slot, she could not indicate the orientation of the slot, either verbally or manually. However, she was as good as healthy volunteers at quickly placing a card in the slot, orienting her hand appropriately from the start of the movement. So now let's move on to the second case. The case of AT, the woman who could not accurately grasp unfamiliar objects that she saw. So AT had a lesion of the occipitoparietal region, which likely interrupts her dorsal route. So the case of AT is in major respects complementary to that of DF. Okay? AT is a woman with a lesion, again, to the occipitoparietal region, which interrupts her dorsal route. AT was able to recognize objects and demonstrate their size with her fingers. In contrast, the pre-shape of her hand during object-directed movements was incorrect. So as a consequence, she could not pick up objects between her fingers. Instead, the patient made awkward palmar grasps. So yes, AT could describe what she could see, but when she was asked to pick them up na when she was asked to interact with the objects or at least grab the objects. She could not do so. She could not adjust her hand to reach for the object. She only made awkward hand or palmar movements or palmar grasps. Okay? Although AT could not pre-shape her hand to pick up neutral objects like blocks, when she was presented with a familiar object of a standard size like a lipstick, she grasped it with reasonable accuracy. So again, this is like another heavy implication of the control of behavior versus conscious perception theory, that the dorsal stream specializes in visually guided behavior and not just spatial perception of things. So yung nagagrab lang ni AT, yung mga at least familiar objects of standard size like the lipstick, but she could not grasp unfamiliar objects that she saw. Now let's move on to the two areas of the secondary visual cortex. So the two streams, the dorsal and ventral stream, do not function in isolation from one another. It has been suggested that they are also composed of two substreams. One is the fusiform area in the ventral stream, and two is the empty or the middle temporal in the dorsal stream. So, andito daw yung empty. And the fusiform area is here. It's not visible on ventral surface of temporal lobe. So we have neuropsychological disorders linked to damage by the substreams. So in the fusiform area and the ventral stream, we have what we call prosopagnosia. Okay? So first, para mas maintindihan natin ang word, agnosia means a failure of recognition. So visual agnosia naman is a specific agnosia for visual stimuli. So prosopagnosia is a visual agnosia for faces that can be acquired either during development, which is known as developmental prosopagnosia, or as a result of brain injury, otherwise known as acquired prosopagnosia. So prosopagnosia is a visual agnostics with a specific difficulty in recognizing faces. And even in extreme cases, prosopagnostics cannot recognize themselves. So now let's try to understand the brain pathology of prosopagnosia. So diagnosis of acquired prosopagnosia is often associated with damage to the ventral surface of the brain at the boundary between occipital and temporal lobes. This area of human cortex has become known as the fusiform face area because parts of it are selectively activated by human faces. Okay, keep that in mind. It's known as fusiform face area because parts of it are selectively activated by human faces. Lastly, because electrical stimulation of this brain area in humans can metamorphose a viewed face into a completely different face. The belief that prosopagnosia is a deficit specific to the recognition of faces has been challenged. Careful testing suggests that some prosopagnostic patients have a general problem recognizing specific objects that belong to a complex class of objects. For example, particular automobiles or particular houses, and not just a specific problem recognizing faces. Let's dive deeper into this, okay? So, prosopagnosia is typically applied to neuropsychological patients, diba, who have difficulty recognizing particular faces, but they can readily identify other test objects like a chair, a dog, or a tree. So you'd think that this is powerful evidence, diba, that prosopagnostics have recognition difficulties specific to faces because they can identify that is a chair, but they can't identify specific faces. But however, this is not the case though, according to other studies, because prosopagnostics have no difficulty recognizing faces as faces. The fact that they could recognize chairs as chairs, pencils and as pencils, and doors as doors is not relevant, because prosopagnostics are aware that they are looking at the face, at the human face at least, or at any face for that matter, a dog's face or something. They can identify that that what they are looking at is a face, they can identify what they are looking at as a chair. So the critical question is whether they can recognize which chair, which pencil, and which door, and most importantly, which face they are looking at. So careful testing of this sort usually reveals that their recognition deficits are not restricted to faces only. So there was this one case, for example, a farmer who lost his ability to recognize particular cows. A farmer lost his ability to recognize particular cows when he lost his ability to recognize faces. So in another patient or in another study, with routine testing, RP, the name of the patient, displayed a severe deficit in recognizing faces and in identifying facial expressions, but no other obvious recognition problems. So if testing had stopped there, as it often does, it would have been concluded that RP is an agnostic with recognition problems specific to human faces. Because yun lang yung tinest eh. Yun lang yung na-observe. He had deficit in recognizing faces and identifying facial expressions, and yung iba, the other recognition problems were okay daw. But more thorough testing indicated that RP is deficient in recognizing all objects with complex curved surfaces and not just faces. So there is more of a specificity in prosopagnosia. It's what they're trying to understand more, that prosopagnosia is not restricted only to the deficit in recognizing faces as faces. So there are more factors that contribute to the unrecognition of, in this case, human faces. So there are many more. Hindi lang sa mga mukha naghihirap ang mga prosopagnosia patients. Conscious awareness in prosopagnosics. So, the fact that prosopagnosia results from bilateral damage to the ventral stream suggests that dorsal stream function may still be intact. For example, the control of behavior versus conscious perception theory suggests that prosopagnosics may be able to unconsciously recognize faces they cannot recognize consciously. And this is indeed the case. So Chanel and Damasio in 1985 were the first to demonstrate that prosopagnosics can recognize faces in the absence of conscious awareness. So they presented a series of photographs to several patients. Some photographs were familiar to the patients and some were not. The patients claimed not to recognize any of the faces. However, when familiar faces were presented, the subjects displayed a large skin conductance response. So a large skin conductance response is a kind of bodily, how do I explain this? Bodily reaction, something related to the skin. Para siyang micro reaction in a way, but this is tested in a machine, something like that, if I'm not mistaken. So again, they had a response which did not occur with unfamiliar faces. So thus, indicating that the faces were being unconsciously recognized by the undamaged portions of the brain. Amazing. So now let's move on to the second neuropsychological disorder linked to the two streams. So we have Akinetopsia. This is in the empty area in the dorsal stream. MT is also known as V5 or MT-V5. Akinetopsia is a deficiency in the ability to see movement progress in a normal smooth fashion. This can also be triggered by high doses of certain antidepressants, and akinetopsia is associated with damage to the MT area or the middle temporal area of the cortex, where its function appears to be the perception of motion. So if the fusiform area was responsible for activation of specific faces, empty area or the middle temporal area, its function now appears to be the perception of motion. 95% of the neurons of MT respond to specific directions of movement and little else. Also, each empty neuron has a large binocular receptive field, allowing it to track movement over a wide range. So as mentioned, Akinatopsia is or may be triggered by high doses of certain antidepressants. So drug-induced Akinatopsia, we have two cases here. First, a 47-year-old depressed male receiving 100 milligrams of nifazodone twice daily reported a bizarre derangement of motion perception. Each moving object was followed by a trail of multiple freeze-frame images, which disappeared once the motion ceased. And the second case, a 48-year-old female receiving 400 milligrams of nifazodone once daily at bedtime, reported similar symptoms with persistent multiple strobe-like trails following moving objects. So in both cases, stationary elements were perceived normally, indicating a selective impairment of the visual perception of motion. Vision returned to normal in both patients once the dosage was removed.
Finding segments!
So that ends chapter 6, the visual system. Thank you very much for staying with us in our report. This has been Group 1, the Visionaries. So if you guys have any questions, please do not hesitate to ask us, and we will try our best to answer them. Thank you and have a good day.